Hello, Professor Akshit. Yeah, we are ready from our side.
Okay.
I think Anugrah, I think sir is ready to go, right?
Yeah. We can start now. Yeah, sure.
All right. Great. Well, it is a great pleasure to introduce Professor Christian Schlotterer, who is with us from Christian did his PhD at the LMU in Munich, Germany, after which he moved on to do a postdoc with Brian Charlesworth at the University of Chicago. And post that, he moved to Vienna, which is where he is now, at the University of Vienna, where he also founded and heads the Institute for Population Genetics. Christian's work has been very interesting and particularly pivotal in understanding experimental evolution and applying population genetics to it, in particular to populations of Drosophila, which is fruit flies. I'm very curious to hear what he has to tell us about experimental evolution today. Please, Christian.
Good. I have a question. Nobody can ask me questions. Is this correct? I don't see anybody. So they cannot respond. Is this correct?
I believe the AV and the outreach team can help with that.
No, because I was thinking to ask questions, but I don't see anybody in Zoom, just us. And that's why it's a bit okay, good.
Actually, we'll take up the questions from YouTube. They'll post the questions in YouTube comment section and we'll take it up from there.
Okay, but it's not possible to have it interactive. That's sorry, this was just my misconception, but we'll manage. Okay, good. I will share my screen. Okay, so as has been already announced, I will be talking about experimental evolution. And if we talk about experimental evolution, we need first to discuss what is experiment, what is an experiment, and what is actually evolution. I'm sure you all have some ideas about this, but nevertheless, I like to go a bit more into defining the things. So if we ask what is an experiment, and then we know that the scientific experiment belongs to an entire process known as the scientific method. And this method has a set of ordered steps that we are basically using to acquire new knowledge. And I've given you here the steps. So we make an observation. This is just by maybe coincidental. And then we want to find out more about this. And therefore, we ask a question and formulate a hypothesis. And then scientific experiments are going to set up to test this hypothesis and to collect data. Then we will, after we collected the data, we will examine the data and the results. And based on this, we will either accept or reject the hypothesis that we've been formulating in step 2. And at the final step, we basically report the results, and scientists typically do this in scientific publications. And these publications are then peer-reviewed, and after colleagues have said this is a solid result, it will be published and made available to the community or a broader audience. So an experiment is a procedure designed to test the hypothesis as part of the scientific method. There are 2 key variables in any experiment that are independent and dependent variables. The independent variable is controlled or changed to test its effect on a dependent variable. So, for example, we have the hypothesis that temperature of water determines how much sugar can be dissolved. So I'm asking you all now, despite I cannot hear your answer, you should name what is the dependent variable in this experiment and what is the independent variable. Good. I'm not going to resolve it. You will have sleepless nights. So as we now have a vague idea of what an experiment is, we need to know the second component, and what is evolution. And evolution has been defined by a famous evolutionary biologist, Douglas Futuyma, as the change in the properties of groups of organisms over the course of generations. It embraces everything from slight changes in the proportions of different forms of a gene within a population to alterations that lead to the earliest organisms, to dinosaurs, bees, oaks, and humans. So it's a very broad definition, and it encompasses quite different things. So let's try to be a bit more specific. I've given you here 3 examples. This is the first example, and what I'm asking you now, do you think evolution has occurred from this form to this form, from the top form to the bottom form, and here also from the top to the bottom? So I would love to see your hands raised. Who thinks this is evolution? Obviously, they differ dramatically. So everybody, I think the majority of you will probably assume that there has been evolution that changed this form into this one into this one, and this one into this one. But of course, life is not as easy. In fact, these are genetically identical organisms. And the only reason why they differ is that they have been raised under different environmental conditions. So the environment can affect the phenotype, as we say, the physical appearance of an organism without any genetic change. So we would not call this evolution, but rather the technical term for this is called phenotypic plasticity. And this is something very, very important and interesting because it makes it sometimes very difficult to distinguish between a plastic response, this means an environmentally triggered change or a variation, and a genetic response, where the genes are responsible for the change in phenotype. So what we need for evolution is a change that is heritable. It is something that is carved in the genome. Therefore, it's a genetic basis. What evolution is, this is basically changes in allele frequencies. So now again, I'm asking you, do you know what an allele is? And then this time I'm going to answer it. So you can think that if you have a genome and there is a certain specific locus in this genome, and this locus can have different forms. You can compare it like a switch. A switch will be something like a gene, and then you can have different positions. It could be on, off, or up, down, or whatever. And this state of on, off, up, down, this is an allele. And the switch corresponds to a gene. So that's the difference. We call the position in the genome a gene, and the variant that we find there is called an allele. And so what we need in order to have evolution, we need to have genetic variation. Without variation, there is no evolution. And what happens during evolution, that these variants are changing in their frequencies. One variant may go up, the other variant may go down. And this is the essence of evolution. What is of interest? Whether the evolution is happening by neutral forces or by selection. And of course, we are interested more in selection than by neutral forces. But you wouldn't believe it, it's in fact not easy to distinguish between neutral and selective forces. So, as we can have selective forces and neutral forces, it makes it very hard to find out whether an outcome that we see today has been driven just by chance processes or by directed forces. And therefore, Stephen Jay Gould has had this fantastic idea. Wouldn't it be great if we could rewind the tape of life, basically going back and going through the same procedure again? And what he's writing, you press the rewind button and making sure you thoroughly erase everything that actually happened, go back to any time and place in the past, then let the tape run again and see if the repetition looks at all like the original. If each replay strongly resembles life's actual path, then we must conclude that what really happened pretty much had to occur. So there must be a force that drives this. But suppose that the experimental versions all yield sensible results, strikingly different from the actual history of life. So again, I have a question and I don't know, and I would love to see what you would think. If we think we replay the tape of life, going back to the beginning of life on the planet, would we basically have humans being on the planet again or no? Think about this. I'm very curious. I would love to hear your answer. I have no clue, if I'm honest. I don't know whether it would look the same, and I cannot answer it. But it's a very interesting thought experiment that you think about, if we play it again, how would it look if we had the opportunity? And this is, of course, a fascinating idea. And it has been kind of taken on the idea in the film movies. And I guess you're all too young to remember the Groundhog Day. This was a very popular movie when I was young. There a person wakes up every day. The day starts exactly the same. And then he can modify the behavior and see what happens. But on the next day, basically, he starts from scratch again. Another version of this is a French movie that I really love, and it's called Smoking, Non-Smoking. And there, it's always a path of life, and you make binary decisions. Either you take the left door or the right door, and then the movie goes back. What would have happened in life if you would not have taken the left door, but the right door? And then you would see that your life would branch out to completely different fates on your history or fates of your life. You would have been a very different person. It can depend whether you take the left door, right door, whether you smoke a cigarette, whether you don't smoke the cigarette. Very little decisions in life could potentially have a big impact on your long-term projection, how your life, career path, life path will happen. So it's a beautiful movie. Unfortunately, you can't barely get it any longer. Has not been very popular, I think, but I love it. So if you have any opportunity, look at Smoking, Non-Smoking. But let's go back to science, what we think about how we can address this. And can we do this? And this is where experimental evolution comes into play. With experimental evolution, we have more or less the opportunity to rewind the tape of life. But typically we do not in a sequential manner, basically playing it again and again and again, because we're impatient. We're scientists. We want to have the results fast, and we want to know the answer very quickly. We play the tape in life, not again and again, but we play it in parallel many times. So what you can see here is basically each line is a evolutionary trajectory. And what it shows is the frequency of a variant. It's a frequency of an allele, what I've been saying to you. And what you can see is that we have changes in allele frequency. And according to the definition we used before, if you look at this lineage has clearly evolved because the frequency has changed. One allele has gone up in frequency. The same here, one allele has decreased in frequency. We have evolution. But the problem with this is all the experiments look different. One, I've shown you the two extreme ones where we see the frequency increase or frequency decrease of the same allele, and many of them, they just meander around. And such a pattern is nothing else than a random pattern of evolution, just something that happens by chance. And this is what we call as population geneticists, genetic drift, but it's nothing else than sampling variation. So it means without selection, we can have evolution, but it is not directed. So every realization, every replaying of the tape gives you a different result. And this is the reflection of chance. So it's clearly interesting. And for evolutionary biologists, it's very interesting also to study the impact of chance. But on the other hand, yeah, if we know it can be this or this, and both is equally likely, that's not super exciting. What we are more interested in, what are directed changes? And these directed changes we call selection. And this is what you see in the right panel here. Again, each of the gray lines is one realization. And sorry, I forgot to say this, the yellow one is the average. So now what you see, we have a directional force operating selection. And selection is operating towards increase of allele frequency. So you see on average, in all of the experiments, frequency increases. And now we have a directive force. We can see the replaying the tape of life gives us a similar answer. Frequency increases, but we also notice there's still a large stochastic component involved. And what I said, that the challenge for evolutionary biology is to distinguish between genetic drift and selection. If we have only a single observation, it's very, very difficult, because we can see this frequency change here is as high with selection as this frequency change here under neutrality. So the same outcome is possible under selection as well under genetic drift or under neutrality, random forces. But it also shows you now the power of replaying the tape of life, because if you do not have a single observation only, as we typically have in nature, if we can replay the type of life, if we can make the same experiment multiple times, then by doing it multiple times, we get a very, very convincing answer, because we see overall all the experiments go up. Under neutrality, 50% go up, 50% go down, and then we can distinguish between selection and neutrality. And this is one of the strong power of experimental evolution, that we can replicate experiments, and this facilitates the distinction between neutral forces and selection. So the next thing is, we have to think about how fast is evolution. Is evolution sufficiently fast that we as experimenters can observe it? Because it makes no sense to make experimental evolution if evolution is so slow that we cannot follow it in our lifetime. And the answer is, fortunately, turns out evolution can be quite fast, which contradicts some of the assumptions that evolutionary biologists had for a very long time. And there are a couple of examples that you may be familiar with where evolution is happening fast. Probably the most prominent one that I'm sure all of you have heard about this is antibiotic resistance. And this is evolution in action. We are basically trying to kill bacteria by taking antibiotics. And of course, bacteria don't like this, so they are experiencing a strong selection pressure to find a way to detoxify or make the antibiotic useless. And this results then in antibiotic resistance. So this is evolution in action. And in this aspect, we don't consider it very good that it happens, but it happens. Another example is also for herbicide resistance. It's very well known that when you spray herbicides, plants or the herbs basically develop resistance against the herbicide, and then it's pretty useless. And this resistance typically happens by novel mutations. And then we can also have resistance against pest control. For example, we try to control rats by putting warfarin there, and it's complex how it works, but it's quite obvious it doesn't take long, and the rats develop resistance against warfarin, and then it's no longer as effective. So typically, when we try to make a massive killing of a bacterium or some pests, we see that resistance develops very, very fast. And this is encouraging. Well, it's bad for us in this context, but it's encouraging for experimental evolution because it means, yes, we can do it, and we should be able to see evolution in the lifetime of a researcher or a lifetime of a grant sometimes. I would like to give you another example of a very well-known case of rapid evolution, and this is the peppered moth. You may know the story. The peppered moth in its original form has been sitting here on a tree bark that is covered with lichen. So, and the lichen is very light in color, and therefore, if you have a light color on the moth, it's perfectly camouflaged during the day. And why is this important? Because during the day, predators like to eat them. So, in principle, this is a perfect adaptation. Camouflage on the white background perfectly masks the moth. What has happened in the UK and other parts in Europe, that industrialization resulted in quite a strong pollution of the air. And these lichen are super, super sensitive to air quality. And therefore, when the air was getting polluted, we see the bark of the tree, but now it's free of lichen. And now what happens? We can see here the moth. It's perfectly visible. And this was creating a very, very strong selection pressure, because now all the birds could see the moth that was sitting there during the day, and they easily found their yummy meal. So there was strong selection for the moth to camouflage again. And now how would you camouflage in the dark bark? Well, you just turn dark. And this is what you see here. This is now a camouflaged organism. It's very difficult to see. And here in contrast, you see the dark form here. And this has happened quite fast. You can see basically the frequency of the black moth was quite low. And then around 1900, it has basically almost all of the moths were dark. What you can see here, that the frequency of the black one is going down again. I ask you, can you think or a reason why this is the case? It is because air quality has improved, and therefore lichen were coming back, and therefore the camouflage, the dark camouflage was no longer effective, and this one was getting better again, and therefore the frequency of the black one is going down. A very beautiful example of rapid evolution. The cool thing is we even know now what is the driver of this. This is that a mobile genetic element, we call it a transposable element, has been inserting in the proximity of this gene, and this resulted in the change in the color. It's a beautiful story. We have a history of the change in phenotype across time, and we know even the molecular basis of it. A beautiful case study of rapid evolution. Just to show you that people are becoming more aware that rapid evolution is something quite widespread. If you look at a number of publications mentioning contemporary evolution or rapid evolution, you really see that this has been widely recognized in the scientific community, that evolution can be rapid, and therefore it's worthwhile talking about this. So we have all the ingredients. We know what experimental evolution is. We know why experimental evolution can be cool. And we know that we should see evolution in action when we run an experiment. So let's see what we can do. So during experimental evolution, we study populations across generations. And this is important because we need mutations to occur or allele frequencies to change. And this can only happen across generations. The advantage of experimental evolution is that we have defined reproducible conditions, which is different to the wild. We can do this in the laboratory or nature, but in nature, of course, the conditions are more difficult to control. We can do replication. I think this is a key factor, and I've shown you this in the beginning, how important this can be. And we can also include control populations, which is very important. So we can have a treatment So, for example, we can treat the population of bacteria with antibiotics, and we have an untreated one, and they come and ask, how do they behave differently? That's a perfect setup we can do. So the oldest experimental evolution study that has been done is basically dating back to 1856. It's called the park grass experiment. And you can see this, and you can see here on the figure several plots. You can see there is an area, and in this area, there have been basically, it's a hay meadow, it's a meadow for producing hay, and they were asking what is the impact if we use different types of fertilizers. And you can see each of these square is one of the treatments where a certain type of fertilization method has been used. And you can see already from this graph that this results in different growth behavior of the grass. And so that's a nice experiment. We can ask what happens there. Then we can ask how's the species composition changing, many things, how's yield changing. All these questions can be asked. It's the longest experimental evolution study that I'm aware of that has been running. I would like to introduce you another experimental evolution study that has basically almost all world records. And I was indicating this in my abstract as well. And this is a long-term experimental evolution study. It's with E. coli. In case you don't know what E. coli is, E. coli is a bacterium, Escherichia coli. It's a gut bacterium that you all have in your gut. It can be good, it can be bad. So some of them are pathogenic because they have some genes that make you sick. But typically the lab E. coli is very nice, very friendly. You can drink it if you want. It doesn't do any harm. It's good, no problem. And you can easily cultivate it. And this experiment was conceptually super simple. You basically start from a single colony. This is a plate on which these bacteria are growing. And this is a colony. And as this colony is isolated, you know that it has a single bacterium there. And then this bacterium was growing, multiplying, and this is what you have, a colony, or yeah. So if you start your experiment by inoculating a set of parallel cultures, he's been using Richard Lenski as the driver behind this, and he's been using these flasks here. So you inoculate a series of flasks with the same descendants of a single bacterium, and then you just let it go. So they basically can be traced back, all the parallel cultures that you can see here. They can all be traced back to the same ancestral clone. And then they have been continued evolution. So this goes until whatever 2020, and it has been more than 65,000 generations. This is fantastic. And in principle, the point they make, it has been evolving so long that every position in the genome had the opportunity to mutate. Now we cannot discuss. I cannot discuss anything with you, but it's a simple perspective, and I will show you that this is not as simple as you may think, that any mutation can happen at any time. But it's the longest experiment in terms of number of generations that we ever had. And this is the guy that initiated this. He's Richard Lenski, and he has been doing this for more than 25 years. So he basically provides a cost, how much this has been costing. It's actually quite cost effective because he has done many publications, and his students have been continuing with experiments. There's a real family of scientists that all go back to the lines that Rich Lenski has been creating. So he had really big impact by starting this very, very simple experiment. It's very simple. And it's also what I should say, it's essentially hypothesis free, contrary to what I was saying, that an experiment tests an hypothesis. In principle, the idea is just, the hypothesis is, if I have a bacterium, I put it in a lab environment, it will adapt to the environment. This is the simple hypothesis, but he has then been able, and I will share some of the results with you, to come up with a very interesting series of experiments that tell us a lot about adaptation and evolution. So one of the first insights that we got from this study, and this is a very short period, it only covers 2,000 generations, he could show that there is adaptation. And at that time, sequencing was way unthinkable. So you could not sequence bacteria to find whether they had a positive mutation or not. So he was thinking about a way, how can I show that there has been a beneficial mutation occurring, because initially all the cells are identical. And as I told you, we need variation for selection to operate. And in these type of experiments, the new mutations are not present at the beginning, but they occur during the experiment. So every cell division, you may have roughly 1 mutation per cell. And so you get mutations every cell division. As you have many, many different cells there, you can generate quite a bit of variation in a relatively short time. And so what he was doing, in order to say, has my population adapted? He was always comparing the evolved population. Evolved means having been cultured for several generations, and comparing it against the ancestor. And this is very easy what you can do in E. coli. You can freeze them in the freezer, and then you can always go back. It's also very cool with E. coli and some other organisms like Caenorhabditis, where you can freeze them. And then you can always go back to the past and ask, well, how has been this historic sample? How did it behave? And he's doing this. He goes back to the founder genotype and competes it against the evolved population. And if the evolved population has acquired beneficial mutations, you would say that the evolved population outcompetes the ancestral one. So they have more offspring in the same amount of time. And this is what he was measuring. He calls it here the relative fitness, because it is the ancestral population as the benchmark. He always compares it, and then he compares evolved populations at different points in time. So this is maybe a population that has evolved, whatever, 250 generations. And what you can see here is a very interesting pattern. He finds that the fitness remains relatively stable. Then it jumps up, remains stable, jumps up, remains stable, jumps up, and then remains stable again. So the interpretation that he had between this generation, a beneficial mutation must have occurred that was then spreading through the population and conveyed a fitness advantage. So this population is not as fit as this one, and therefore you see the jump here. And this happens again here. Somewhere there it happens a new mutation, and this new mutation gives us a fitness advantage. We can see again a big jump in fitness. This population is much fitter relative to the ancestor than this one. And so here he could basically track when he thought that the beneficial mutation has occurred in this experiment. I can tell you now, it's a very clever way, and it was very informative, but it grossly underestimates the number of mutations, beneficial mutations that have occurred, because it's a very coarse assay. So if we do now whole genome sequencing of these populations, you can do this now. We have these, I don't know whether you've heard of this, there's called this next generation sequencing that allows us at very low cost to sequence entire genomes in many copies. It has been really a breakthrough in biology. And very interestingly, I should say, it's a breakthrough in biology, but what has been causing this breakthrough is basically engineering, because people were able to design a machine that can do the sequencing with very small volumes, and you don't need a lot of chemistry and all this. So it's automation that basically made this so affordable. But it's a breakthrough in biology. So I try to explain to you what you see here. So you see here, this is on the x-axis, we see time. And on the y-axis, this axis here, we see frequency. And then we depict different alleles in different colors. For example, if you focus on this one here, you can see that this allele has been a very low frequency, and then it shoots up in very short time until it becomes fixed. This is a typical signature of a beneficial mutation that has occurred. And you can see this multiple times, one beneficial, another beneficial, another beneficial, another beneficial, another beneficial. That's a very striking pattern. So when we do this, we can see a series of interesting insights. So we see that many mutations, initially at low frequency, but then rise in frequency until they go to fixation. This is the classic case what we think happens to a beneficial mutation. A beneficial mutation occurs, gives an advantage to the bug, and then it goes up in frequency, competes all the other bacteria, outcompetes the other bacteria until only the descendants of this one that have a beneficial mutation are in the population. So this is clear, and there's no big surprise. But what was observed as well, what we see here, a mutation occurs, it goes up, so it suggests it's beneficial, but then this mutation goes down again. Here we see the same. Goes up, disappears. Goes up, disappears. This seems to be a very common pattern. Here, for example, this one. You see, this goes up almost to 40%. It's a very, very high frequency. And then it disappears again. The other interesting pattern, what we see here, some mutations hang around for many generations. For example, if you look this, there are many mutations, they stay here and more or less go zigzag, go a bit up, go a bit down, go a bit up, go a bit down. It's a completely different trajectory compared to the one that we saw here, that we were thinking, well, this is what we expect to see. And also what we find is the number of mutations that we see along time differs over time and lineages. So how can we explain this, and what do we learn about evolution from this? I first would like to introduce to you a concept that's called clonal interference, and this gives the explanation why a beneficial mutation can go up and still go down. So if you think that this is basically, again, on the x-axis, we have time. On the y-axis, it gives some frequency measures. And so we start here. This is the entire population. So the frequency is 100. And then in this population, a beneficial mutation occurs. As we've been saying, the beneficial mutation spreads in the population. And this is what we see here. The beneficial mutation spreads. It becomes this A area becomes larger and larger and larger. But as the population size is so large and mutations are quite frequent, what can happen, while the A mutation is on its way to fixation, there's still a large population remaining that doesn't have the A. And so it could be that in this population without the A, another beneficial mutation occurs, and let's call it C. And so what this C allele is doing, it's also better than the ancestral population. So it's outcompeting the ancestor, and therefore the C allele is increasing in frequency. But it turns out the A allele is better than the C allele. So this is very good, and this one is just good. So this can increase to some extent, but then when it starts competing with the A, it has been outcompeted. And this is what we call clonal interference, because they are clones. Each bacterium is a clone. They're not exchanging genetic material. And therefore, the clones are interfering with each other, and that's why we call it clonal interference. The stronger mutation A replaces the beneficial mutation C and the beneficial mutation B. If the A mutation would not have occurred, it is quite likely that either A, B, or C would have become to fixation. And this shows you also some of the random factors that can happen in evolution, because it depends. The fate of C depends on whether you have the A or you don't have the A. If the A is not there, C will be fixed, and all bacteria would have the C allele. But in this case, we don't have it. And this pattern can explain why the frequency goes up for a short period of time of C, and then it disappears because the A is taken over. So we've explained the first miracle that we saw there. So I would like to give you a short detour because I think it's so interesting. It is so interesting. The question, have you ever thought about why we're having sex? Why sex is so widespread? And sex just don't blush. When evolutionary biologists talk about sex, they mean basically recombination, which happens during sexual reproduction. This means that you basically have the possibility of exchanging genetic material in the offspring, and yeah, you exchange genetic material between the father and the mother in the descendants of the offspring. So this is basically the opportunity where you can have recombination. And so the situation that I was explaining to you is only applies to clones when we have no sex, when we have the mutations A, B, and C. What would happen if we would have sex? In the case of recombination, these lineages are not competing against each other, but what you can do is you can actually create an organism that does not have only the A allele, but also have the A allele and the C allele and the B allele. And then very quickly we generate a very good organism by combining the beneficial alleles all into this one individual, and this is the winner that takes it all. In a sexual population like E. coli, this can happen as well, but there first A needs to fix, and the new mutation needs to occur on the background of A. Then we first have AB, and then we need to have, if AB is fixed, we have a new mutation where A, B, and C are then on the same genotype. And so what we can see is that the advantage of sex is that it can combine favorable alleles very quickly, what you cannot do in asexual population. And this is one of the explanations, but it's not a satisfactory one. I don't want to go into deep depth, but it shows you this is one of the thoughts why having sex equals recombination can be beneficial for adaptive processes. And we can look at this. So we predict that populations having sex when adaptation is really important, they could have an advantage. So this has been done in a study where people were making an experiment competing sexual versus asexual yeast population. This is cool. With yeast, you can have it with sex and without sex. So you can really compare yeast, the same yeast cell in the presence of sex or without sex. And they were comparing these populations in a benign environment where not much stress is being imposed and a very stressful environment. And so what you can see is that the sexual population is in here. This is in the case where we have a benign environment. I don't know. One is the sexual, the other one is the asexual. You see that sex makes no difference. So if the environment is good, we don't need many beneficial mutations. Doesn't matter whether we have sex or not. But if we have a stressful environment, you can see that the fitness, this is basically how good you are, is indicated on the y-axis, is significantly better when you're having sex than we have no sex. So this is a nice illustration, empirical illustration of the theory that I was explaining to you, why sex can be beneficial. So let's go back to our experimental study. Again, this was, of course, a test also done with experimental evolution. So then the second thing I was explaining to you is that some mutations hang around for many generations, as we can see this. They don't go to fixation. This is also not compatible with the very simple idea, because if we have a beneficial allele, it should go to fixation. So how can we explain this? And the idea is a process that's called cross-feeding. In other words, that some bacteria are living on the leftovers of others. This means that you could have bacterium A metabolizes something like glucose, and then there are some byproducts from eating glucose that can be still have energy, But this bacterium is not using it because it just eats glucose or whatever, saccharose, whatever. And the leftovers that are not being consumed by this bacteria can be eaten by other bacteria. And so they basically are feeding on the leftovers of the other bacteria. And this is what you call cross-feeding. And so they basically can both persist because they don't compete with each other. It's not that they're out-competing each other. And so we can see this. There are two genotypes, and they're basically, you can ask them, so you can start with different frequencies. This is the ratio of the two genotypes. One where the one is very common, and here is the other very common, and then you let them evolve. And you see that independent of the ratio of the two genotypes, they go basically to the same equilibrium. And this is indicating that there is this evolutionary stable state when we certainly have a certain ratio of these two genotypes, because this is what is done. One feeds glucose, the other one eats on the leftovers, and so they have a stable equilibrium, because the one that eats on the leftovers is dependent on the leftovers of the first one. And so that's how you get a stable equilibrium. So this could explain what we are seeing in this experiment. Something what's very, very interesting is also what has been seen in this experiment, that, and it is related to a bit to this, is that the ancestral E. coli cannot really metabolize citrate. And so if you go through the experiment, you see there is basically, they can't metabolize citrate, but at a certain time point, suddenly they can metabolize it. So they can use citrate as a carbon source, basically as a food, and this was not possible here. So you may scratch your head. Christian has been telling you their population sizes are so large, the experiment is so long, that basically every mutation could have occurred at any time point. So then you may ask, well, if every mutation is possible, why hasn't it been possible that this mutation has occurred already much earlier? Because citrate has been there, you could have used it, and it would have been good. And so there can be two different explanations. The first one is maybe the idea is not correct, that every mutation could have happened at any time. In other words, this is a very, very rare mutation. Or the other explanation is a very complicated one. It says basically it's historical contingency. It means, in other words, that if you have this mutation at early generations, it's not enough, this mutation, to generate the ability to metabolize citrate. So, so basically what it means that you need to have prepared the cell with other mutations, that this mutation actually can result in the citrate metabolizing observation, whatever, property. So what you see here, they did an experiment to ask basically, what do we find? So they measure the mutation rate, and one is the mutation rate. In red is the mutation rate to generate the mutation for generating citrate, and the other one is for background, a mutation in another metabolizing enzyme. So what you find here, if you use the ancestor, you see, okay, we find with a certain rate this mutation for metabolizing the substance. But we don't find a mutation for citrate. But then they use clones from around this point in time, and they call them potentiated because they have background mutations that are being needed. And when they do this, wow, now they can see that we have basically the possibility to get it. So there's a very big difference in the mutation rate in the ancestor and the potentiated clones. But this mutation rate is only for the metabolization for citrate. For the arabinose, this other substance, there is no difference. So it's not that the mutation rate in general has been changing. It only has been possible to generate a mutation that allows the metabolization to citrate when other factors have been already mutating before. So this is what we call historic contingency. Whether or not a mutation has an effect depends on what other mutations are in the background. And this is also important for understanding cancer, for example. Whether or not a certain mutation results in cancer depends to some extent also on your genetic background. Whether you can metabolize a drug depends on your genetic background. So a lot of the properties of a mutation are not just sitting in the mutation, but also depend on a genetic background. And this is a beautiful example for this. And the last thing I like to discuss with you on this beautiful experiment is that the density of mutations differs over time in lineages. So what you see here is basically a large number of clones that have been sequenced, and you ask what is their relationship to each other. And then you basically plot a phylogenetic tree that shows you the number of substitutions. The longer this axis is, the more mutations have occurred. This means that they've all been sampled at the same point in time, but you find that some of them, the majority, has not many mutations. And there are some exceptions, for example, like in this lineage here, there are many mutations have occurred because these branches are very, very long. And the conclusion is that in this lineage, there has been a change in mutation rate. Suddenly, there has been a mutation been fixed in some individuals that result in a higher mutation rate. And where does the higher mutation rate come from? This is that basically DNA repair is being inactivated. Another aspect of historic contingency is that E. coli strains can evolve to the same optimum when they have different background. So initially, they are not equally good. And when you evolve them, then they become a very similar phenotype to this. So this is on the phenotypic level. It means that historic contingencies means what genetic background they had was not important for doing this on the phenotypic level, but remember what we've seen for the citrate. So it depends on a case by case. Another important thing for historic contingency is antibiotic resistance. You need 5 mutations to basically get from wild type to a E. coli that has antibiotic resistance. And so we can basically run experimental evolution, we can acquire random mutations, and then we can ask, is the order of mutations actually important for getting this? And to make a long story short, yes, the order of mutations is very important. So you show the thickness of the lines, tells you basically the frequency at which mutations are occurring, and you can see the resistance. And the long story is basically there are only a few paths that evolution can take that really lead you to a resistant cell. And therefore, it's again historic contingency. If you go in the wrong direction, you basically end up with a non-resistance. So this, if you go this path here, there's not a good chance that you end up there. This is a case of historic contingency. You're more likely to acquire full resistance if you go this path with the solid lines. Another interesting observation is that adaptation is not a one-way street. It can be reversed. And so you can do experimental evolution, not just with bacteria, but you can also use bacteriophages. These are like small viruses for bacteria. And these viruses can be adapted to their host. And you can use as a host either Salmonella or the E. coli that we've been talking about already. And so if you basically have this phage adapted to the host Salmonella, then it cannot grow on the E. coli host. But if you have it adapted to E. coli, then it doesn't affect the growth on Salmonella. And so what they did is an experiment where they basically measure it in in one condition, and then they change the condition. So this is in Salmonella, then you change it to E. coli, and then you back to Salmonella, and then you back to E. coli. So what you can see here, what you can see is this effect. So you maintain it in the cells or the phages on selection on Salmonella, you see that the phage acquires, I should basically, acquires mutations, 3 mutations. And then when they have the adaptation to Salmonella, they cannot grow very well on E. coli anymore. Then you shift it back to E. coli. And now what is happening? Selection is operating in the opposite direction. This frequency of these new mutations goes down again. And you can see that then the phage can grow again very well on E. coli. So in one regime, you acquire mutations that make you fitter in this environment. And then you change the environment or the host from Salmonella to C. elegans. And then you see that these mutations are being reverted. And the reversion is that new mutations occur. Phage has a very high mutation rate. So you can back mutate very easily. And then you end up with the same sequence that you had before. It's really cool to show that depending on the host, either mutations occur or they disappear again. Very cool what you can do. Okay, so I see that I've been a bit slow. I would like to have some feedback. How much time do I still have? Hello? Okay, good. Then I just continue. With now experimental evolution without crossing species, not with bacteria. The challenges with outcrossing species are that the population sizes that we can have are much smaller. We have longer generation times. So the experiments are more difficult to do and they take longer. But the very interesting difference is that adaptation does in these experiments not occur by de novo mutations, but because populations are too small, so you do basically adaptation from standing genetic variation. You use adaptation from a natural population, and therefore new mutations are not as important. Our favorite organism is, isn't it beautiful? This is a fruit fly, Drosophila melanogaster, and you can see a female, which you can recognize on the pigmentation, the absence of sex combs. And what I would like to talk about, something that you cannot do in yeast or E. coli, is to study basically the war between the sexes. And what's very interesting, that males and females have different requirements. So you would think males and females are part of the same species. They should basically all need the same thing. This is not correct. So what happens if you have basically a mutation that's beneficial to the male but bad to the female? This cannot really spread in a population, because if it's good for the male fitness increases, but female fitness decreases. And basically, as we have sex every generation, this is being shuffled, variants have been shuffled between males and females. So indirect evidence for sexual conflict comes from the seminal fluid, which reduces the competitive ability of the sperm of other males, but has toxic effects to the females. So basically, we have the seminal fluid. This is basically sperm. The ejaculate does not only contain sperm, but also these accessory gland proteins, and they are needed for fertilization, and they can be used by the males to basically compete for fertilization success. And what they do is basically by reducing the successful fertilization by other males after this male has mated, but this is not good for the female, and these proteins have toxic effects on the female. So basically, by mating, the male injects sperm and something that kills the female. It's a very interesting property. And so there has been a beautiful study that really did the painstaking work to quantify the fitness of an allele in males and females. And so what you can see here, in blue lines, we have genotypes that have a very high fitness in the male, but a very low fitness in the female. And vice versa, there are some genotypes that are not very good in males, but super good in females. And then you have some genotypes with something in between. But the interesting thing is, it's this allele that's very good in the male will never make it to fix in the population because it's not good for the female. Maybe we skip this. So what is the hypothesis? In the case of sexual conflict, the trait cannot evolve because if it's what is good for the male and bad for the female, it can never fix. And the hypothesis has been that to resolve this conflict is very difficult because we need to move the genes around in the genome. We need to find complicated mechanisms to basically prevent the cost. And what you could have, it either could be benefit only one sex, but no cost in the other, or the benefit to both sexes. This would be one way how you could resolve the conflict. So we did an experiment with using fruit flies with experimental evolution. We have an ancestral founder population, which is now polymorphic. It's a natural population. So the variation is there. And we generate 10 replicates. And then we can basically maintain these replicates for 100 generations. So we may transfer them every generation. And then we have 10 evolved replicates, and we can ask, how do they look like? And so what we looked at the evolution of gene expression, we asked, how is gene expression changing? And so we measured the change in gene expression in the female and in the male. So we have each basically a line, and we ask, what happens to gene expression? If gene expression is evolving, this means probably it was good. But keep in mind what I said before, what's good in the male can only increase if it doesn't harm the female, or it's also good in the female. If it's good in male and females, we should see the gene expression changing in both sexes. If it's only good for one sex, it should either not change, or if there's no cost associated, it may change in both sexes, in only one sex. And so what we find here is in rat, you find a lot of genes that change gene expression only in the female. And in blue, we have genes that are changing gene expression only in the male. And now this is really shocking because it means that the change in gene expression has only benefit to one sex, but not the other, because we see it sex restricted. And the question is, why is this the case? How could this happen? This is just a zoom in that we can see sex-specific evolution for lipids. There's basically a reduction in the lipid content for the females. We don't see any change in the males. We see sex-specific evolution for neural signaling. There are octopamine-related genes that are changing only in the females. We see dopamine-related genes only changing in the males. So there's all neural signaling, and the neural signaling is evolving, and this is very sex-specific. So we see this octopamine is related to egg laying. It makes sense that the evolved females lay more eggs than the others. And this one has to do with basically activity in the hot environment. So basically what I conclude is that sex-specific adaptation is rapid. And what we think is that why this can happen contrary to the expectations of antagonistic sexual selection is that there are some regulatory factors, and we call them transcription factors. So some transcription factors are having a sex-specific gene expression. And if these genes that are changing in a sex-specific manner are regulated by the sex-specific transcription factors, there's no cost. Because if this gene is only expressed in the male and not in the female, or the change in expression is only happening in the male and not in the female, there's no cost. There's only the benefit for the males, but no costs for the females. So what we are concluding is that this idea of the conflict between the sexes is certainly true, but there are many mechanisms that allow a rapid adaptation of sex-specific traits without causing harm to the other. And I think I stop here. I would have had another story for you about ecological speciation, but I think for the sake of time, I better stop here. So, and I'm happy to take questions if you have any. Hello? Um, hello? I've been told I should talk an hour. Sorry, can you give me feedback? I would love to answer some questions, but okay, good. Then I continue further. Good, then let's do ecological speciation. I really like to have some feedback. Okay, good. So then let's continue with ecological speciation, which I think is a fascinating story as well. I'm happy to share it with you. So there is the idea is that if you have ecological speciation, we can have adaptation to different environments. And so this is the key idea that populations adapt to a specific environment by genetic changes. And these genetic changes do not only provide an advantage in the new environment, but they also cause incompatibilities between individuals from the adapted population compared to a population that is not adapted, or even a population that is adapted to a different environment. So you can have populations either adapting to hot and compare them to an ancestor, or you can compare a hot population to a cold population. And then the idea is they adapt to the hot and cold environment, and as a side product, they are becoming no longer compatible with each other. And therefore, reproductive isolation between populations from different environments builds up as a consequence of adaptation to a certain environment. So there is no direct selection for reproductive isolation. This is just a neutral byproduct by the effects of the genes involved in adaptation to different environments. What's very important prediction is that replicate populations from the same environment show no reproductive isolation. So you only find reproductive isolation between populations that are adapted to different environments, but not independently evolved populations that are adapted to the same environment. So we ran this experiment and we were asking, what do we find? So we were looking at basically pre-mating reproductive isolation. We did two different ways. We were measuring with a video device where we were basically exposing females and males together, and we're asking how well did they like to mate. And we did different comparisons. We matched ancestral males and ancestral females, or we matched hot evolved males and hot evolved females. And then we were basically using the video, and then you can use KI basically to track the individuals and ask, well, how much time does a male spend courting the female? And what we find is that the ancestral males do not spend very much time courting, but the hot evolved males, they spend a lot of time courting the female. So there has been evolution that the males spend more time courting the female. Good. But this doesn't say anything about reproductive isolation. So we made basically mismatched. So this is the ancestral female with the the ancestral male with the evolved female, and this is the hot evolved female with the ancestral male. And what we can see in this comparison is that the mating activity or the courting activity dramatically dropped, irrespective of whether we had this contrast or this contrast. In both cases, when we have a mismatch it means the male is less interested courting the female. So this shows some way of reproductive isolation, that the male is not interested in courting with the female. So you may be saying, well, okay, this is not so unexpected. They come from very different populations. Maybe they're not used to this and they had no choice. So let's do it better, give them a choice. And that's what we did. We basically made this experiment here when we gave them a choice that each female could either choose between ancestral male or a male that was hot evolved. And so then we find that when we combine hot evolved and ancestral males and females, we see that they prefer to mate with each other rather than with the wrong ones. And this doesn't matter which of the replicates we're using. It's always that ancestral males prefer to mate with ancestral females, and evolved females were preferring to mate with evolved males. If we mix different replicates, and remember the prediction that replicate populations do not evolve in parallel, basically, or evolve in parallel and do not develop reproductive isolation, and this is exactly what we see. They don't prefer each other. If anything, then they prefer the other one. So there is no evidence for reproductive isolation when we test among evolved replicates, only when we find, make a comparison between ancestral and evolved ones. So there are many adaptive responses in hot temperature. We find in neural signaling evolution, as I've been showing to you, or the lipid metabolism. And the lipid metabolism is very interesting because the enzymes that are involved in the cuticular hydrocarbon synthesis are also part of the lipid metabolism. And everybody who's familiar with Drosophila immediately blinks the red light when you see CHCs, because this may indicate mate discrimination, because it has been widely documented that changes in the CHC composition can lead to mate discrimination and reproductive isolation. So what we measured is a CHC composition, and you see there's a big difference as a principal component analysis. You can compare on the PC1, you find the differences between males and females. This is not very surprising, has been described a lot, that males and females have a different CHC composition. But what's very interesting is in the second principal component, you can see here that the evolved ones are the solid circles, the ancestral are the empty circles, and you can see that in both sexes we find a very parallel response, is that namely that the CHCs have been changing in the ancestral, in the evolved populations relative to the ancestral one. And you can then break it down by using the loadings on the PCs, and you can see that basically almost all CHCs are affected to a larger or lesser extent in the changes in evolution. And then we can basically complement this by doing gene expression analysis. We can ask which genes are changing expression, just as we've done it before. And then we see that there's a very consistent pattern in gene expression changes that we see in the genes that are involved in the synthesis of the cuticular hydrocarbons, just confirming the pattern that we've seen, that the CHC composition changes, and the genes involved in synthesizing them have also changed their expression pattern during evolution. And so the conclusion is that we basically find patterns of reproductive isolation, which is pre-mating, before they actually mate, which fits the pattern of ecological speciation. So, but it's very important that this reproductive isolation is a byproduct of adaptation to lipid metabolism, which is probably a response in terms of the adaptation to the hot environment, but it's not a direct target of selection. So reproductive isolation has occurred, but only as a side product, because the populations needed to adapt to the hot environment. For this it was important to change the lipid metabolism, and the change in lipid metabolism as a side product resulted in this reproductive isolation. And it's just a summary if we find reproductive isolation between the ancestor and the evolved ones, but not between the different evolved replicates. So I stop here. I thank you for your attention. If there is nobody here, I have no questions, then I will just shut the Zoom and end my session. I'm sorry for this. Yes. Okay, I wish you all a lovely day. We have a holiday here in Vienna, and I hope you have learned something. And then I wish you all a very nice remaining day. Greetings from Austria. I hope you enjoyed the detour into experimental evolution, and you've seen that this is a very powerful tool, and I hope you get excited and like to learn more about this. Thank you very much, and I will stop the recording. So I don't know. I find this very frustrating that there is no feedback. There is no possibility. I'm speaking and I have no clue what you're saying. Sorry, this is super frustrating. Anyway, so I will shut my computer down and I'm sorry for this. Bye-bye.